What is recombination DNA? Recombination DNA, known as a technology that uses enzyme to cut and paste together DNA sequence. Recombination is a process by which pieces of DNA are broken and recombined to produce new combination of alleles. But here's the question: how does recombination DNA work? DNA recombination involves the exchange of genetic material, either between multiple chromosomes or between different regions of the same chromosome. In this case, a sister chromatin serves as a donor of the missing material. Recombination occurs when two molecules of DNA exchange pieces of their genetic material within each other. An example of DNA recombination. For example, insulin is produced by means of recombination DNA with bacteria. A human insulin gene is introduced into plasmid, which is then introduced to a bacteria cell. The bacteria will then use its cellular machinery to produce the protein insulin, which can be collected and distributed to patients. The first step is DNA isolation. DNA isolation is a process of purification of DNA from sample using a combination of physical and chemical methods. As you can see in the diagram, there is a DNA isolation and the DNA purification. DNA isolation contains from cell lysis, protein removal, DNA precipitation, and DNA rehydration. These are the steps of DNA isolation. And there is the DNA purification, sample precipitation, DNA binding, wash, and DNA eluation. Creation of lysate. The first step in any nucleic acid purification reaction is releasing the DNA RNA into solution. Its goal is to completely distribute cells in a sample to release nucleic acid into lysate. Second step of recombinant DNA is cutting of DNA enzyme digestion. Process of cutting DNA molecule into smaller pieces with a special enzyme called a resurrection enzyme. Resurrection digestion is the process of cutting DNA molecules into smaller forms with a special enzyme called resurrection enzyme. Let's call it RE. Resurrection digests begin by mixing the DNA and the RE. The RE are kept on ice until the producer begins. Why? Variables pH, NaCl, concentrations are optimized by adding specific buffers of the enzyme being used. And here we go for the third step, amplifying of the DNA. Recombinant DNA technology. Here, as you can see in front of you, the gene of interest. Then, the gene of interest is amplified by the process of PCR, that is, the polymerase chain reaction. The polymerase chain reaction is a specific technique in which very multiple copies of gene of interest are synthesized into version. Requirements of PCR. First, DNA template, nucleotide primers, and enzyme. Let's begin with the DNA template. Template is a target of DNA that needs to be amplified. And the primer, it contains primer. As you can see in front of you, primer are complementary to the strands of the DNA or the DNA template taken. Enzymes are proteins that help speed up metabolism or a chemical reaction in our bodies. They build some substance and break other down. Requirements of the PCR target DNA primer and DNA polymerase. Steps of the DNA chain reaction: denaturation, annealing, and extension or elongation. Let's begin with denaturation. It is a target DNA heated at high temperature during the process of denaturation. In biology, it means process that modifies the structure of protein. It involves the breaking of many of the weak linkage or bonds. As example, hydrogen bonds. Annealing means when the temperature is lowered to enable the DNA primers to attach the template DNA. Two primers are added during annealing at optimal temperature. Annealing when temperature is lowered to enable the DNA primers to attach to the template DNA. Extending when the temperature is raised and the new strand of DNA is made by the tag polymerase enzyme. The fourth step is joining DNA. If two DNA molecules have matching ends, they can be joined by enzyme DNA ligase. DNA ligase seals the gap between the molecules, forming a single piece of DNA. But here comes the question. How are the two DNA fragments joined together? Two fragments of the DNA may be joined by DNA ligase, which catalyze the formation of a bond between three at one end of the strand of DNA and the 5' phosphate group of another. A nick in the DNA, for example, a break in one strand of a double standard DNA, can be repaired very effectively by the ligase. Then to the fifth step, insertion of rDNA into a host. The insertion of recombinant DNA into a host cell. Preparation of insertion of recombinant DNA into a host cell. A vector DNA and a foreign DNA carrying gene of interest are cut by the same resurrection enzyme. DNA ligase enzyme forms a new sugar phosphate bonds to DNAs. The ligated DNA is introduced to the host cell. Here, as you see, the gene of interest and the ligase and the vector DNA ligase work as a glue. As the host cell replicates, recombinant molecules are based on tubronoline. Clone DNA can be recovered, analyzed, can be transcribed, or translated in a host cell. The ligated DNA introduced to a host cell, the behavior of the host organism change. Here the resistance genes that marked as purple enter the E. coli cell. And finally, the final step, recombinant cell isolation. We see four tubes, let's mark them, A, B, C, D. A, letter B, cells are treated with prostrate to destroy protein. C, cells be released in situation. Supernatural liquid containing the DNA is transferred to a clean tube. And the last, which is D, the DNA is precipitated with its form strands that can be on a glass rod.